everyone. I am Dr. Divya Jain, Department of Botany, ST College, Ambala, Kent. In the previous tutorial, I discussed with you some types of DNA damage and also told you that the cells have evolved mechanisms to repair this damage. In this tutorial, we will look at some of these repair mechanisms. Now, what actually is a DNA repair mechanism? DNA repair is a collection of processes by which a cell identifies and corrects the damage to its DNA molecule. You see, the cells they have evolved certain proteins that sense the damage to the DNA and then recruits the DNA repair machinery to repair that particular damage. Just a quick recap of the previous tutorial. This diagram shows different types of DNA damage that we have already discussed in the previous tutorial. For example, mismatches or deamination and depurination of bases. We also discussed oxidation of the bases by the free radicals. We discussed alkylating agents. We discussed bulky adducts. We discussed single strand and the double strand breaks. Along with all these modifications of DNA, we also discussed certain exogenous and endogenous factors that cause this type of modification or damage to the DNA molecule. Now, students, the interesting part is that the cells have not only evolved various sensor proteins that sense the DNA damage, but also they have evolved certain mechanisms such as mismatch repair, base excision repair, nucleotide excision repair, and many more DNA repair mechanisms to repair the damage of the DNA. In this particular tutorial, we will focus on some of these repair mechanisms. Now, I will discuss all the mechanisms stepwise. Focus on the diagrams which are self-explanatory. I have written down the steps for you in the slides as well, so it would be easier for you to make notes and follow the steps of the repair mechanisms. The first two mechanisms that I'm going to discuss involve the direct reversal of DNA damage. The first is photoreactivation. Direct reversal of DNA damage is with the help of certain enzymes which directly repair the damage to the DNA molecule. Remember to focus on the diagrams. So this first mechanism is photoreactivation. As you can see, exposure of DNA to UV radiations, it causes two adjacent thymine bases that are existing in the DNA strand to become covalently linked together. So that means there are two adjacent thymine bases and they become covalently linked together because of the exposure to UV radiations. Now, this dimer, it creates distortion or it produces kinks in the molecule of DNA, and this kink or this distortion, it affects the replication as well as the transcription. Let's see how the replication is disrupted by the thymine dimer. Now, as the polymerase enzyme, it is replicating the strand of DNA with thymine dimer. The polymerase enzyme, it does not recognize the thymine dimer and it can't replicate past the thymine dimer. That means this dimer, it serves as a kind of a speed breaker for the polymerase enzyme. The polymerase enzyme, it separates from the template and again rejoins the template, and where it has separated, that means at this position where it has separated, it would leave a gap in the daughter strand. Now, a gap means a deletion in the DNA strand. The cells, they have evolved a mechanism known as photoreactivation to repair this thymine dimer. Let's see how. I told you that this is a direct repair. An enzyme photolyase has been evolved in the cells, and this enzyme, it becomes active as the thymine dimer is formed within the DNA strand. This enzyme photolyase is a light dependent enzyme. It becomes active, it detects the thymine dimer and binds to the thymine dimer. Now, what it uses the light energy to break the bond between two adjacent thymine bases. That means the bond between the two thymine that has caused the formation of the dimer, it is broken by the enzyme photolyase. That means this enzyme, it catalyzes the reaction which breaks the bond between the two thymine dimers, and therefore the damage has been repaired. Remember one thing, that the photolyse works only in the presence of visible light, especially the blue region of the spectrum. And without energy derived from the visible light, photolyse cannot catalyze the cleavage of bonds of thymine dimers. So the enzyme photolyse, it breaks the two covalently linked thymine bases. This photolyase enzyme has also been found to break the cytosine dimers as well as the cytosine and thymine dimers in the single strand. The second mechanism of direct reversal of DNA damage works on alkylated bases. An enzyme known as alkyl transferase, it removes the methyl or ethyl group from the bases. We have already studied that when the bases, they become alkylated, the DNA becomes damaged. But luckily, there is an enzyme, alkyl transferase, that removes the methyl or the ethyl group from the nitrogenous base by transferring the methyl or ethyl group to its own cysteine side chain. So the alkyl transferase enzyme has a cysteine side chain, and the alkyl group is shifted to the side chain. Okay, so now the base has been repaired, but one very unique process happens over here. That is, when the alkyl group of the base is transferred to the enzyme alkyl transferase, the enzyme becomes permanently inactive. Now, this is not something the enzymes usually do. These enzymes, they are again available for further action, but this enzyme alkyl transferase does not do so. Once it receives the alkyl group, it becomes permanently active, and therefore it is known as a suicide enzyme. You see, in a way, it actually sacrifices itself while repairing the alkylated base of DNA. So, hope you understand what is the direct reversal of DNA damage. That it is the repair of the damage directly by an enzyme. So, we have discussed two enzymes in these two processes. One was for reactivation, where the photolyase enzyme was active, and another is alkyl transferase enzyme. Now, I'm going to tell you about three mechanisms which follow cut, copy, and paste. You all must have used this mechanism at least once. The first mechanism of this type that I'm going to tell you about is base excision repair mechanism. As the name indicates, the mechanism involves excision. Excision means removal or cutting. excision or removal of a damaged base from the DNA, and that damaged base is replaced by the normal base. And the mechanism, it follows the principles of copy sorry cut, copy and paste. Let's see how. The mechanism of base excision repair mechanism is initiated by an enzyme glycosylase. Focus on the diagram, students. Now, like I just told you, base excision repair is initiated by an enzyme glycosylase. But one thing you should be clear about is that the specific type of abnormal base is recognized by a specific glycosylase. Here, I have taken the example of a cytosine base which has deaminated to uracil. Okay, so cytosine deaminates to uracil, maybe due to exposure of certain external factors, or maybe due to some endogenous factors, or sometimes spontaneously also cytosine deaminates to uracil. Uracil, as you know, is not the normal base of DNA. And if it is not removed and replaced, it can cause mutations from GC base pair to AT base pair. This process we have already discussed in the previous tutorial. Now, let us see how the base excision repair mechanism takes place to remove this uracil from the DNA strand and replace it with a correct base. And this mechanism, it works on the principle of cut, copy and paste. Now see how it works. Like I just told you, the enzyme glycosylase recognizes the abnormal base. Here in this case it is uracil. Now the enzyme glycosylase, as it recognizes the uracil, it binds to it and cleaves the glycosidic bond between the abnormal base and the deoxyribose sugar, creating an AP site. You know that the deoxyribose sugar is covalently linked to the nitrogenous base by means of glycosidic bond. This glycosylase enzyme, it cleaves the glycosidic bond between the sugar and the base, and it creates a site where there is no nitrogenous base, and that site is known as the AP site. The AP site is recognized by enzymes endonucleases and phosphodiesterases, and together endonuclease as well as phosphodiesterase, they cut or excise the sugar phosphate groups at the site where there is no base present, and that is what is the cut part of the mechanism. DNA polymerase then replaces this missing nucleotide. You see here the base was first removed and then the phosphate group and the deoxyribose sugar was also removed. Now this gap, it is recognized by DNA polymerase and it replaces the missing nucleotide using the undamaged complementary strand of DNA as a template. This will read the template and it will insert the correct base, and this is the copy part, and the cut ends, they are sealed by the DNA ligase, and that is the paste part. So this works on the principle of cut, copy and paste. Let's see another example of this mechanism, that is nucleotide excision repair mechanism. As the name indicates, here few nucleotides are excised or removed along with the nucleotide carrying the abnormal base, and then all these nucleotides, they are replaced. So there is a abnormal base, and this mechanism, it removes a few nucleotides, and those few nucleotides, they include the nucleotide that is carrying the abnormal base. So let's see how cut, copy, and paste works over here. This mechanism rec- recognizes and removes thymine dimers. Thymine dimers, as you know, are two covalently linked thymine bases in the same DNA strand. And like discussed earlier, these are usually formed due to exposure to UV light. We have already discussed one mechanism that repairs the thymine dimer, that is photoreactivation. This is another mechanism that repairs the thymine dimer. The mechanism is based on excision activity of an enzyme Xe nuclease that produces cuts on either side of thymine dimer. So this this is the cut part. You see, this is the thymine dimer. Xe nuclease activity will make a cut on either side, this side as well as the this side, and then this piece of DNA molecule, it would be removed and replaced. Let's see how. I will be discussing this mechanism in E. coli. In E. coli, nuclease activity requires the product of three genes. And the product of genes, you know, they are polypeptides. So there are three polypeptides, UVRA, UVRB, and UVRC. Let's see how these polypeptides they work. 2 UVRA and 1 UVRB polypeptide recognizes the defect in the DNA molecule, that is the thymine dimer, and binds to it. So 2 UVRA polypeptides and 1 UVRB, it binds to the damage and utilizes the energy from ATP to bend the DNA at the damaged site. Now UVRA is released and UVRC protein binds to the UVRP and the DNA complex. Now see what happens. UVRB protein, it cleaves the phosphodiester bond 5 nucleotides away from the third prime end. So UVRB would produce a cut in the DNA chain on the third prime site, and usually it is the fifth phosphodiester bond from the damaged site. UVRC protein, it cleaves the phosphodiester bond from the fifth prime site, and it is usually the eighth phosphodiester bond from the damaged site. So both UVRB as well as the UVRC, they make cuts in the damaged strand. B from the third prime site, C from the fifth prime site. Now you see here the cuts they have been made, and this portion of DNA is separated from rest of the DNA molecule. There is another protein which is UVRD, which has the helicase activity. This UVRD, then it releases or it removes this excised short nucleotide chain, which is around of 13 nucleotides, and now UVRC is also released. DNA polymerase 1 now comes into action and also removes the UVRB. Now you can see over here B is also removed, and UVRD has also removed this small piece of DNA carrying the damage. As the B gets removed, DNA polymerase 1 then reads the complementary strand and fills the gap with the correct bases. So now this was the cut part of the mechanism where B and C they have made a cut, and this is where the polymerase 1 is reading the strand, that is the copy mechanism, and when the DNA ligase it seals the cut ends, it is the paste part. So polymerase will read the complementary strand, fill in the gap, and ligase will seal the end, and the thymine dimer has been repaired. So here are the notes that are written for you. You can just read them and understand the concept. The next cut, copy, paste mechanism that I'm going to discuss is mismatch repair mechanism. And as the name indicates, it repairs the base pair match, that is mismatch in a DNA molecule. So it repairs the base pair mismatch in a DNA molecule, and that is why it is known as mismatch repair mechanism. It also works on cut, copy and paste mechanism. Let's study the mechanism with the help of a mismatch example. Suppose during DNA replication, T mispairs with G instead of A. So that the thymine usually base pairs with adenine. And if it base pairs with guanine, then it is a mismatch. Now, interesting part over here is that though it is a mismatch, but both the bases are normal bases of DNA. दोनों ही DNA के normal bases हैं. So now for an enzyme that is sensing a mismatch, it is very confusing which base to remove as both the bases are normal to DNA. The question is how to identify the older template strand where the mismatch is located. You see, most of the mismatches they happen during replication. So it is the daughter strand that would be carrying the mismatch, and the template strand would be a normal strand. But for an enzyme, it cannot identify whether G is the mismatch or it is the T is the mismatch. So enzyme is confused. So the question is how to identify the older strand from the new strand. Well, students, the cells they have a solution over here. The template strand is identified from the new strand with mismatch by the presence of methyl group on the template strand. You see, there is an enzyme called DAM methylase or DAM methylase that adds methyl groups to the adenines in this sequence GATC sequences of DNA. The methyl group is added to DAM in the template strand. And as the newly formed strand, it is not methylated because it will take time for them to add methyl groups to the daughter strands. So mismatch repair system uses this difference in methylation to remove the mismatched nucleotides in the new strand, because now it can know that okay, this base I have to remove because that particular strand is unmethylated, it does not carry the methyl group. The repair system cuts the mismatch and replaces it with correct nucleotides using methylated parent strand as a template. So in a nutshell is the mechanism is that the enzymes that are involved in mismatch repair, they have to identify which base is to be removed from the strand, because both the template strand as well as the replicated strand, even though it is a mismatch, both are carrying the normal bases. So it is the newly formed strand which is carrying the abnormal base or a mismatch. So enzyme has to recognize that which is the template strand and which is the daughter strand, and it uses the methyl pattern to recognize it. The template strand would be methylated and the newly formed strand is unmethylated, and that is what is recognized by the enzyme. Now let's see the repair process now. Three proteins designated as MUT-S, MUT-L, and MUT-H, they detect the mismatch. So here there is a mismatch where G has mispaired with T. This yellow one is the template strand, and as you can see it is methylated, and the blue one is the new strand, and for the time being it is unmethylated. And these genes, they just recognize this newly formed strand and know that they have to repair T instead of G by this methylating pattern, and they bind over here. Now see, mut H. Mut H, it identifies the methylated strand and binds there. Mut S locates the mismatch. So there are three genes I told you, or three proteins that are the products of three genes: Mut S, Mut L, and Mut H. So the Mut H identifies the methylated strand and it binds over there at CATG site, and Mut S, it locates the mismatch, and once it is detective, the mut S, it forms a complex with mut L. You see, mut L, as you can see, it acts as a linker between mut S and mut H, and due to this complex formation, now the DNA molecule, it forms a loop. Once the mut S, mut L, and mut H complex is formed, this mut H, it makes a cut. It makes a cut in the unmethylated new DNA strand. So mut H, it will make a cut in this blue strand, and after the strand is cut, here the cut is made, after the strand is cut, mut U, it's not shown over here, mut U, it just separates the two strands, and now an enzyme exonuclease 1, it will remove the nucleotides from the newly formed non-methylated strand in the direction of the mismatch. So it will start, it will make a cut over here by the mut H will make a cut over here, and then cut, and it will remove the nucleotides from the new non-methylated strand in the direction of the mismatch, and it proceeds beyond mut S. And as I told you earlier, mut S is binded to the mismatch. So the cut would proceed beyond the mut S protein, and the mismatch would be removed. So that was the cut part, and DNA polymerase 3 now comes into action. It reads the template strand, fills in the correct bases, that is the copy part, and the ends they are sealed by DNA ligase, and that is the paste part, and mismatch is now repaired. So hopefully now the cut copy paste mechanisms are clear to you, and also that how the enzymes they recognize which base to remove. We have discussed the repair of a dimer, a mismatch, and an alkylated base. Let us see how the cells repair single strand and double strand breaks. First, let us see how the cells repair single strand breaks. This mechanism has been studied in human cells. When there is a break in the DNA strand, as shown here, the exposed strand is coated with proteins that are called as PARP1 proteins. And these proteins they protect the damage and also recruit the repair machinery. And then after this has been protected, now DNA polymerase comes into action and it fills the gap reading the template strand, and the ends they are sealed by DNA ligase. Double strand breaks of DNA are repaired by a mechanism known as non-homologous end joining, or NHEJ. During non-homologous end joining, the two broken ends of DNA are simply joined back together. This mechanism requires the participation of several proteins. First, the double-stranded break is recognized by end binding proteins known as Ku proteins. So these yellow ones, they are the Ku proteins. They bind to the broken ends of DNA molecule. These proteins then they recognize additional proteins, and these additional proteins also join the complex, and they form a cross bridge between the two DNA molecules, and these proteins which have formed a cross bridge they prevent the two ends of the DNA molecule to drift away from each other. That means they do not allow that these two broken molecules they should separate from each other. Then some more DNA processing proteins are recruited in this region, and these proteins they process the ends of the DNA molecule, and finally if there are gaps that are repaired by DNA polymerase and the ends they are sealed together by the enzyme ligase. So these proteins they simply stitch back to DNA molecules if they break or if the breaks are in the two strands. However, there is one disadvantage that sometimes there is deletion in this region. Now, if a region of the genome has suffered extensive damage, that means if the DNA has suffered extensive damage, then the repair mechanisms are unable to handle the damage. Now the cell faces an emergency and it has a tough choice either to die or to replicate the damaged region, even though this replication may be error prone, and it also results in lot of mutations. अब सेल के पास दो ही चॉइस है, either to die or to replicate the damaged region. So it has been observed in E. coli that when faced with this choice, the bacteria they take the second option. That means the E. coli cells they take the second option, and the second option is of the replication of damaged DNA and induce an emergency procedure which bypasses the sites of major DNA damage, and one such mechanism is SOS response. Now, see, DNA polymerase 3, it is the main replicating enzyme, and this is the portion which is a highly damaged DNA molecule, and this strand of DNA is now being replicated. It is now being replicated, and this is the DNA polymerase 3 enzyme. As it is moving on this template, as soon as it reaches this highly damaged DNA part, it detaches. And as it detaches, this enzyme is replaced by another enzyme which is called as a mutosome. Mutosome is a combination of DNA polymerase 5 and REC-A protein. So this complex is a mutosome. DNA polymerase 5, it has the capacity to replicate the damaged part, and the REC-A protein, it helps the DNA polymerase 5 to replicate this part. It forms a kind of a coating over the damaged polynucleotides, enabling DNA polymerase 5 to carry out the error prone DNA synthesis through the damaged region. And after that, or beyond that, now the DNA polymerase 3 can then take over. So replication has taken place, but there would be lot of mutations in the cell. The cell preferred mutations over death. Now, what happens if the DNA repair mechanisms fail? Now, you see, due to the failure of these mechanisms, the cell cycle becomes irregular. Replication, transcription, and chromosome segregation all show irregular processes, and because of this, failure of mechanisms can cause senescence or aging or programmed cell death, cancer, and inborn metabolic errors. Xeroderma pigmentosum, a type of skin cancer, is the classic example of failure of repair mechanism that repairs DNA damage caused by UV rays. Let's take a quick recap. In the two tutorials, we studied various exogenous and endogenous factors that cause different kinds of DNA damages. And we also discussed the repair mechanisms and the consequences if the repair mechanisms they fail. This is the end of the tutorial. Before you leave, try answering these questions to know how far you have understood the concepts. Define DNA repair. Why have DNA repair mechanisms evolved in nature? Define photoreactivation. Which enzyme is known as suicide enzyme? Explain the base excision repair mechanisms. What is NER mechanism? What is mismatch repair? What is NHEJ? How does SOS response occur? Why is DNA damage repair necessary? These are some references that I have used in making of this tutorial. Students, DNA damage and repair is an interesting topic. I thoroughly enjoyed while making this tutorial for you. Hope you feel the same. Happy learning.